Good morning, everyone here present and present online. So I'll talk you and I'll guide you through some of the key results and some snapshots of all the research around microbial protein that has been going on over the past year, actually. So microbial protein through resource recovery, and I'll also be showing that this is more like a cross-pipeline topic. Actually, the pipelines concerned are water and CO2. Now, what is microbial protein? For those that don't know it, we have a very typical process outline. We start with an inoculum resources, carbon, nitrogen, phosphorus, energy, mixing, etc. We produce microbial biomass in a bioreactor. We then harvest it and do some further downstream processing, which can involve drying or other extraction or other additional steps. Then we have microbial protein. We can use it for food, feed, fertilizer purposes, and depending, of course, on the application, we go for other quality and content and other criteria. Now, this is very important at the international level, at the EU level, but also at the regional level. Flanders has this very nice protein transition strategy, and microbial protein fully fits in it because we want to go for more sustainable protein and we want to go for more novel protein. Now, what do we do in Capture? We do RD&I, and we focus on several domains. So at the core, of course, we have our bioprocess. We have the choice of inoculum and resources, how we bioproduce, under which operational settings, in which reactor configuration, and then how we do the harvesting and downstream processing. So in the middle, you see all the technological choices that we can make, and we do this and we check this with the outcome and the key performance indicators that you can see here. Environmental impact, we want environmental sustainability. We want economic and legal aspects to be covered. We want this to do this in a very cost-efficient way. We do pay attention to legal requirements and procedures, to achievable tonnages and quality and its consistency there. And then at the bottom, of course, you see also a lot of the criteria for the actual bioproduction and the product. We want to produce at high performance. You see a number of indicators like rate, yield that are very important. We want to go for nutrition, and this nutrition should be safe and should provide added value. And there's a whole set or suite of parameters that we should take into account, not only in the lab, not only measure the protein, but also test it in vivo with feed and food trials. And of course, and particularly if we go for food, this product should be palatable and should have very interesting functional properties to use as an ingredient in further preparations. So we're at an interdisciplinary domain with this microbial protein, and actually we're at the edge between the primary clean resources and the secondary resources, and often we'll be overlapping. This of course links not only to the hydrogen economy, the CO2 economy, the bioeconomy, but we fit in all kind of sustainable trends and product developments between clean tech and environmental biotech. Now, what organisms do we talk about? We have roughly 5 classes of interest. We have what we call ordinary heterotrophs at the left. They oxidize organics with oxygen to produce biomass. We can, for instance, use sugar or wastewater COD. Then we have methane oxidizing bacteria, or more general, metillotrophs, so any organism that can grow on C1 compounds. It can be methanol, methane, it can be other compounds. You can also see in the table below, I will not go through the details, but there's a number of companies and products that are getting to the market, so this is a very timely moment. Then we have hydrogen oxidizing bacteria. I'll also show one example there. Avicom, a partner of Capture and R2T, is focusing, among others, on this. Then we have microalgae, and last but not least, the purple bacteria, which can grow in autotrophic and heterotrophic mode. So we can also grow them on hydrogen gas and CO2. Now, as I said, we're interdisciplinary and inter or cross pipeline. So this is an example on how we can tap into the carbon of the CO2 economy and the CCU schemes with various groups of microbes. We can directly tap into the CO2 or mix it with hydrogen gas, or first use the CO2 and the hydrogen to get to more reduced carbon molecules like methane, methanol, formic acid, etc. And there's many groups of bacteria that can work on these combinations. Now, if we look at water and wastewater, we can also grow microbial protein on these resources. And here you can see an example of 3 groups of microbial protein and their environmental impact if you grow them on wastewater from a potato processing company. Now, here you can see, for instance, that for human health, the 3 types of microbes considered, the first 3 bars, are scoring much better than soybean meal, which is a very typical and conventional feed ingredient. And also at the level of the ecosystems, we score a lot better. At the level of the resources, and more particularly energy, we should be careful and focus particularly on drying steps and the other steps to look into renewable sources of energy. What about the economics? So we did some techno-economical assessments, and here you can see the net present value, and if it is positive, it is worth investing, so to say. You also see at the secondary axis the payback period, and the green bars show our net present values if we are able to sell our product at the price of fish meal. So if we can sell it as fish meal, we're into business. In the red case, it's the price of soybean meal. So soybean meal is cheap on the market. We cannot get a positive net present value if our biomass is only as good as soybean meal. But I will show you later that we can be definitely as good as fish meal, and we can go up to high replacement rates of fish meal, for instance, in aqua feed. So a payback period, it varies according to the operational settings in our study. But for instance, in this case, with a high rate activated sludge process at a quarter of a day sludge retention time, this is giving the best example. Now, then we need to use these products and take them to the field, do some trials to show that they work at least as good as their conventional and less sustainable alternatives. And in the Micronaut project, we did this together with the Ghent University and Avicom, and we wanted to see how good we can use microbial fertilizers to replace common organic fertilizers. And we had very good mineralization and fertilization results for 4 crops: parsley, ryegrass, tomato, and Sophinia, covering a lot of the horticultural and agricultural sectors of interest already. Now, this was fertilizer. Then we go to feed. Several groups performed feeding experiments, and this is one example in which we took wastewater from a brewery, palm, we upgraded to microbial protein, and we feed it to rainbow trout, which is, well, it's a carnivorous fish. Typically, you use a lot of fish meal. And this graph here actually shows at the left a diet that is fully fish meal based. And then we go with 6 variations of microbial protein up to 75% replacement of fish meal. And we have the same specific growth rate, so the same productivity of the fish. And we have the same FCR, feed conversion ratio. So we're as efficient and we can grow as good these fish. So this is a very nice replacement potential. Now, palatability is important: taste, odor, texture, etc. This is one of the examples of the group in Ghent of Corneel Rabai, and it's showing here that we can be very versatile in tuning for odor and therefore probably also for taste of microbial protein. So this is what we can see here, and more details are in the study of Mirsini Sakarica. Another example to grow in the CCU atmosphere, let's say, is to use light hydrogen and CO2. And we investigated 3 types of purple bacteria, and we can see that they can grow relatively rapidly. They outcompete microalgae. In terms of biomass yield, that means amount of product you get per hydrogen invested. You get a lot more than hydrogen oxidizing bacteria. And we have a very good dietary match with human food. We exceed on all levels the quality that is required to feed the humans. Now, protein quality is very important, and here you see a study of the group of Corneel Rabai, and you see the amount of product you need to eat to fulfill the dietary demands. And at the right you see conventional sources, and the lowest conventional source is soybean. And you see at the left in the 2 colors that all these classes of microbes easily can go below. So with minimal quantities of this biomass, you can fulfill your dietary requirements because the essential amino acid profiles are so good. Then this is also from the group of Corneel Rabai. You see some of the concepts here linking up carbon capture with hydrogen economy. And here you can see that we have from 9 kilograms of ultra pure water, we go to 1 kilogram of hydrogen. Then we combine this with the CO2, we get to reduced carbon compounds like formate acetate, methanol, ethanol, and then we go to oxidize this aerobically. We produce a product with a high value, and furthermore, the CO2 that is released and the H2, we can cycle it back and recapture it to combine with hydrogen gas. Now, another example of an interesting project linked to this carbon economy is the Prometheus project, in which Vito is involved. And here, Vito has a very special role to look into advanced process control and tuning of the biomass quality and productivity. So this is together with Calidris Bio and many other partners, a project funded by Catalysti and Flanderswood. And then finally, we have our industrial partner within R2T, which is Avicom. And Avicom has, among others, two very interesting routes to go to microbial protein. One is starting from wastewater, is Valpromic, and the other one, power to protein, is starting from CO2 and hydrogen. And we'll just zoom a little bit into this hydrogen and CO2 story, which is called power to protein. So this is a microbial gas fermentation platform, and you can see how this fits. We use, of course, renewable power first to split the water and then produce the solar. We don't produce solar protein, we produce the protein, and this process has won the label of the Solar Impulse Efficient Solution. Now, if you're hungry for more, this afternoon we have a session with a very interesting title. It's conversion of CO2 and combination with heteroatoms. So if it's about microbial protein, it's specifically about the heteroatoms, nitrogen, etc. And 4 out of the 6 talks this afternoon in the session that Ramon Ganige and I are co-chairing will be about microbial proteins. So hope to see you there. To conclude, microbial protein, we've worked a lot in all the separate groups in Capture, also in companies related and linked to Capture, and we hope in the coming years definitely to collaborate a lot and move this field forward, this cross-pipeline field. So feel free to get in touch with us, and if you'd like to join or learn more about this. Thank you.